Hello, this is Karen O'Hanlon-Court welcoming you to this Bite-Size Bio web seminar, which today is sponsored by QIAGEN. QIAGEN NV, a Netherlands-based holding company, is the leading global provider of sample-to-insight solutions to transform biological materials into valuable molecular insights. QIAGEN sample technologies isolate and process DNA, RNA, and proteins from blood, tissue, and other materials. Assay technologies make these biomolecules visible and ready for analysis. Bioinformatics software and knowledge bases interpret data to report relevant, actionable insights. Automation solutions tie these together in seamless and cost-effective molecular testing workflows. QIAGEN provides these workflows to more than 500,000 customers around the world in molecular diagnostics, human healthcare, applied testing, forensics, veterinary testing and food safety, pharma, pharmaceutical and biotechnology companies, and academia, life sciences research. Today's presentation is titled qPCR and RNA-seq, the Battle of the Strands, and is being presented by Dr. Joan Chang, who is a postdoc at the University of Manchester, UK. Joan completed an honours degree in biochemistry at Imperial College London, UK in 2009. She started her PhD at the Institute of Cancer Research at the University of London, studying the role of an extracellular matrix enzyme, LOXL2, in mediating breast cancer progression through modifications of the extracellular matrix. Halfway through her PhD, she relocated to Copenhagen, Denmark, when her PhD supervisor moved to the Biotech Research and Innovation Centre at the University of Copenhagen. After defending her PhD, Joan completed a 2-month EACR travel fellowship at Karolinska Institute in Sweden, studying the role of reactive oxygen species and their downstream signaling in cancer-associated fibroblasts. Afterwards, she moved to the US for a postdoctoral position at the University of North Carolina at Chapel Hill, where she made several genetically engineered mouse models to study how adipocytes, as well as the damage-associated molecular pattern molecules, HMGB1, are involved in breast cancer and melanoma progression. After an 18-month stay in the US, Joan has now returned to the UK, where she is a postdoctoral research associate at the Wellcome Trust Centre for Cell Matrix Research at the University of Manchester. She is currently studying how the extracellular matrix, in particular collagen 1, is secreted and turned over in fibroblasts. She is also investigating how circadian rhythm controls this process. Apart from being a scientist, Joan is also interested in scientific communication and outreach, having founded a monthly public lecture series, True Talks, in Chapel Hill that is still running, as well as being a STEM ambassador. She also enjoys fencing, playing ultimate frisbee, whiskey, attempting blogging, and keeping herself alive while cycling in Manchester. As always, we will have a question and answer session after the presentation. So please type any questions that you may have into the questions box, which appears on the right-hand side of your screen, and I'll put them to Joan at the end. The recording of the webinar will be available at /bit.ly/qPCR RNA-seq. So now, over to you, Joan, for the presentation.
All right, thank you, Karen, for a lovely introduction. Thank you all for tuning in to today's webinar. I would like to, first of all, thank Bite-Size Bio for the invitation for me to talk to you about qPCR and RNA-seq, and also QIAGEN for sponsoring today's talk. So just a outline of what I'm going to talk to you about today. First of all, I'll be talking to you about the basics of RNA-seq and qPCR, and I will also tell you about the pros and cons of the two different techniques, i.e. when you should use which technique in your kind of experiments. I'll then briefly touch upon how you should validate your RNA-seq results, and indeed if you need to validate your RNA-seq results, and followed by some common issues are always associated with RNA-seq, and I'll give you pointers on how you might be able to resolve them. And finally, I'll also give you some practical tips in terms of how you might be able to start your experiments using both qPCR and also RNA-seq. So what is qPCR and what does it do? So qPCR stands for quantitative polymerase chain reaction, and I'm assuming everyone here today would know what PCR is, so I'm not going to go into too many details of it. Essentially, qPCR is a specialized PCR-based technique that is utilized in a lot of molecular biology specific labs, and usually it's in the 96 or 384 well plate format. Essentially, we will isolate total RNA from your samples, which could be cells or tissues. And from those total RNA, the messenger RNA is specifically used in this case to generate cDNA through reverse transcription. And then these cDNA would then be used as templates for the amplification of the DNA strands during the PCR process. So this PCR process is monitored real time, because after each cycle, we will measure how much DNA samples, double-stranded products there are. And this also allows for quantification, i.e. standing for the Q in the qPCR. So as a side note here, qPCR can also be called QRT, which stands for quantitative real-time PCR. And certain people also call it RT-PCR, which is real-time PCR. But just bear in mind that if you choose to call it RT-PCR, it could also mean reverse transcription PCR. So be a bit careful about which nomenclature you want to use, especially when you're writing up your paper or your thesis. So going to the basics of a qPCR, essentially we have a master mix that is fluorescence-based. So there are fluorophores are present in this master mix, and there are light sources in the real-time PCR machine would then excite the fluorophores are in this PCR mix, which then would bind. So as amplification proceeds, any fluorescent signals that are released by the fluorophores will be captured by the camera that's also present in the machine. So here I've highlighted two kind of generic PCR mixture that you can use for your qPCR. On the left here is a dye-based PCR mix, and here I've decided to use a cyber green, which is a very commonly used PCR master mix. And on the right here is a quencher-based PCR mix. So in general, cyber green or any dye-based PCR mix, the fluorophores are free floating in your master mix, and they are usually, or they're not fluorescent at this stage, but then they would bind to double-stranded products, and then they would then activate and emit the fluorescent signal. So as your PCR goes on, these dyes will bind to the double-stranded products are being produced and emit the signal that is then captured by the camera. On the other hand, the fluorescence quencher probes are seen here on the right. The probe is a stretch of oligos are specific to your gene of interest, and they will bind they will be attached to a fluorophore on the 5' end and a quencher on the 3' end. And due to the proximity of the quencher to the fluorophore in this case, the fluorophore would not be switched on. However, as the 4' binds with the polymerase and starts elongation, the polymerase would then will come closer and closer to the probe, and as they go through it, they will cleave off the fluorophore, and now the free-floating fluorophore can emit a signal that can be detected by the camera. And here is a kind of generic amplification plot of a qPCR reaction. So on the y-axis here is the fluorescence units, and on the x-axis here is the cycle numbers. So essentially, we always have a baseline, so depicted by the black line here. So this is the background fluorescence that's always present in your PCR mixture, or indeed from your plate that you're using. So as PCR amplification starts, there's always a lag phase or initiation phase, I can see here, but where the reaction is not actually very efficient. However, up to a certain threshold, they would then go into an exponential phase, followed by a linear phase, and then a plateau phase. So this threshold line is basically the level of fluorescence at which we can actually accurately, reliably represent the level of transcripts you actually have in your samples. And the PCR cycle at which the samples reach this threshold is usually known as the cycle threshold CT. However, it can also be known as crossing point CP or quantification cycle CQ. So it depends on which software you use to analyze your qPCR. And as a rule of thumb, if your gene of interest has a low CT value, then it generally means that there is a high expression level in your sample, and vice versa. So high CT means a lower expression. So going forward to a very basic guide to how to generate good results for your qPCR. The first thing is to always make sure you have a pure and clean RNA sample to make good cDNA with. So this will avoid the genomic DNA contamination. And in order to avoid the genomic DNA contamination, you should always DNA treat your samples. So this could be right after you isolate your RNA or during your cDNA synthesis. And second thing is to make sure you have good primer design. So with good primer design, you can actually avoid a lot of the noise that can be generated. So you need to make sure the primers are not binding non-specifically to any stretch of the region of your cDNA, and therefore have non-specific implication. And with good primer design, you can also avoid primer dimers. So in this case, it's quite important, especially if you're using a dye-based PCR reaction mix, because primer dimers are also double-stranded oligo species. Therefore, it will bind the cybergreen and emit a fluorescence signal. And third of all, with good primer design, you can even bypass genomic DNA contamination that might have got through to your cDNA sample prep. So this can be bypassed if you design a primer that is exon-exon spanning, because genomic DNA is always exon and intron intersparsed. So going forward to RNA-seq, so in terms of what it is and what it does, so RNA-seq essentially is a type of next-generation sequencing technique, and it's a super high throughput sequencing of the cDNA that's produced from any type of RNA molecules you wish to study. And essentially, we can profile the expression of a particular RNA species. So this could be messenger RNA, long non-coding RNA, or short non-coding RNA, etc. So we can profile these expressions by counting the number of times the transcripts from this has been sequenced. And on top of that, we can also use RNA-seq, the same process, to study multiple RNA species, but then this again depends on whether or not you choose to do your library prep in a certain method. So the basics of RNA-seq, so I found this cartoon on Nature Methods back in 2013, which I think is a very good representation of what RNA-seq is. So essentially, we would take a book from the transcriptome, shred them up into readable sizes, and then basically stitch them back together back in the book in order to understand what is actually written in the book. And there are specific lingos or languages are associated with RNA-seq. So I'm going to go through them with you today here. So the first thing is library prep. So what this means is essentially how you're actually modifying your cDNA, how you're adding, which adapters you're adding to it, what kind of selection you're going through with the RNA species, etc. The second thing is the read length. So essentially, this is asking how many bases are you sequencing from the end of the transcripts. So this can vary from anything from 13 nucleotides up to, I believe, 1,500 nucleotides or more, depending on your machine. And thirdly, there's a sequencing depth. So essentially, how deep are you trying to go with your sample? So how many reads are you trying to generate from your RNA-seq dataset? From your RNA-seq samples. And another thing that you will always hear people ask about is paired or single end reads. So essentially, this is asking whether you're trying to sequence from just one end of your transcript, so the 5' end, or whether you want to sequence from both 5' and 3' ends, so both ends, i.e. paired end. And finally, there's some measurements of abundance, so RPKM, which stands for fragments or reads per kilobase of transcript per million mapped reads. So this is a mouthful. Essentially, it's just a relative measure for how many times your fragments are detected in your sequencing sample. And there's also TPM, which is a transcripts per million, which is essentially another type of relative measure for abundance. So going on to the workflow for qPCR and RNA-seq. So the first step for both techniques is quite similar. So essentially, you have cells or you have tissues. We're extracting total RNA, and then we have all our RNA species that are available to us to be utilized. So in terms of qPCR, we'll then specifically take out the mRNA, which has a poly A tail, and we convert them to cDNA through reverse transcription. And then we will design specific primers that that in this case, the purple primers are detecting only the purple templates, and then we'll perform a PCR amplification and a detection to see how many of the purple templates are present in our total cDNA pool. And in terms of RNA-seq, we will have all these RNA species available for us, and then we can have various different isolation techniques. So in order to select out the RNA species that we're interested in. So for example, we have poly-A selection, which essentially isolates all the messenger RNA from your samples. Or you can do ribodepletion, which gets rid of the most of the ribosomal RNA from a sample. And you can also do size selection, whereby, for example, if you're interested in short non-coding RNA specifically, size selection would be what you want to do. And from these different RNA species that you've isolated, you will then convert them to cDNA. And then you construct the sequencing library by adding adapters to it, which is then which would then be going through PCR amplification and sequencing. So a good practice for both of these techniques is always to make sure your DNAs treat your samples so that you can remove any potential genomic DNA contamination. So when should you use which technique? So basically, it's a list of pros and cons for each technique to give you an idea of like whether you want to actually use qPCR or RNA-seq in your experiment. So the pros of qPCR is essentially it's cheaper, much cheaper than RNA-seq, and so it's more cost effective if you actually already have a predefined list of genes that you want to study. So I would say anything from a few genes to up to like a few dozen genes, then I would actually prefer to use qPCR, especially if you already have a preconceived idea of why you're interested in this particular list of genes. And secondly, the qPCR, because we're designing the primers ourselves, so it might actually be easier to study isoforms of the protein of interest if you design your primers correctly. And we can also start with very low amounts of starting material. So I've actually gone way down, like as low as 20 or even 10 nanograms of RNA in order to generate enough cDNA for my qPCR results before. And because we are only specifically looking at one particular transcript with the qPCR, that means we can also detect known low copy number transcript a lot easier than RNA-seq, where it might be swamped by a lot of other transcripts. So in terms of cons of qPCR, the first thing is that primers are very important. So this in itself may not be bad, but designing a good primer can be very challenging and time-consuming. And validation and also optimization of these primers can also take a long time. And secondly, there might actually have more bias in qPCR depending on the probe that you use. So it's very common to have probe bias that's based on which region of your cDNA it is actually recognizing and amplifying. So going on to the pros of RNA-seq, the first thing is it combines discovery and quantification. So essentially, you're both quantifying your unknown samples. So you don't need to have a preconceived idea. It's basically like a fishing expedition, as we would call it in our lab. And another really good thing about RNA-seq is that once you have good data, so I need to stress, once you have good data, then you can always go back and reanalyze the data when new information comes out. So for example, maybe you have done your RNA-seq and there's a paper that came out a month later saying that, oh, there are different isoforms or mutations are present in this specific gene that you're interested in. And then you can then go back to your RNA-seq data and say, okay, do I see this in my particular treatment samples or my conditions that I'm interested in? And the third thing is obviously it's a very high throughput kind of technique. So you get a lot of data, a lot of information about your transcriptome. And another thing is that we don't necessarily need to have to do studies on species that has the complete sequence genome. So this goes back to the whole discovery thing. You can actually do de novo alignment of your transcripts and actually build up your own transcriptome mapping, basically. And you can also investigate alternative transcripts quite easily, like at a much higher throughput way. So again, this is dependent on whether or not you're using the right package for this type of experiment. So the cons of RNA-seq is obviously it's expensive. So you need to make sure that your biological question is sound. It makes a lot of sense to use this expensive technique. And secondly, RNA-seq requires high quality RNA. So if you're having, so this doesn't mean that if you have RNA that you've got from FFPE tissues or something that you're not sure how good they are, there are still other ways to bypass this issue with RNA quality, but generally speaking, if you want good data, then you need to make sure your quality of your RNA is good enough for that. And there are many decisions that you need to make in terms of RNA-seq. So for example, you need to decide which enrichment process you want to use, how deep you want to go, are you going to do single or paired end sequencing, etc., etc. So it might be quite overwhelming for beginners. And it's because the amount of data you can get or amount of information you get from one single RNA-seq experiment is a lot. So this makes the data analysis not very straightforward. And unless you have a biometrician, it might be very daunting to try and actually do the data analysis yourself and go through the whole process in order to generate a list of differentially expressed genes for experiments. So the need for a good bioinformatician also goes back to the whole kind of expensive type of thing, expensive concept. So in terms of validation, it's still kind of a little bit contentious because technically speaking, RNA-seq does not have probe bias. So qPCR validation is not as necessary as in validating previous high throughput methods, for example, microarray, because microarrays have a probe bias that's inbuilt to the technique. But you can only bypass this whole validation using qPCR if you have multiple biological repeats that actually have correlation between each of them. So if you only, let's say, have 2 RNA-seq results, then ideally you want to pick a few genes and do your validation with qPCR because it's not powerful enough to answer the question. But there has been a lot of papers out there that shows that qPCR and RNA-seq results are usually very well correlated. And if there are any biases in the RNA-seq, for example, they're always related to the GC content of your sequences. And also maybe you use random hexamers or oligo-DTs in your CDN prep. Or there might be bias towards 3' end or bias towards specific RNA species. And these biases usually arise from the way you prepare your library. So these are all inbuilt, and this can be corrected using computational algorithm at the later stage. So essentially, qPCR is great to validate your 1 or 2 sets of RNA-seq data using multiple other biological repeats. And as I said before, RNA-seq studies are usually quite underpowered to answer the biological question being asked because of the expenses of it. So ideally, you will want to do RNA-seq on, let's say, 20 different biological samples. However, you probably only have enough money for 2 sets of those, and then you will use the remaining 18 and use qPCR validation to look at your hypothesis that's generated from RNA-seq results. So going forward to a roadmap for RNA-seq analysis. So if I've taken this snapshot of this roadmap from this paper in Genome Biology, which I found very, very good for people who are trying to get into RNA-seq analysis. And this link to the paper is actually available at the end of this presentation in the tools slide. So essentially, this is a software roadmap. I've decided to change it into a software roadmap, and I need to clarify that I am a biologist. I'm not a bioinformatician, so I can only give you suggestions on what I know is a commonly used software for each of these steps. So we would first of all have the experiment design and the sequencing design. So essentially, you're deciding whether or not you want to go single, paired end, read length, and etc., how many replicates you want to do, etc. And then once you start having the sequencing data, you would want to perform some quality control. So the sequencing data, depending on which platform you use, you have two different softwares that you can use to read your results. So there's FastQC that is specifically for Illumina, and there's NGS QC, which is any platform. And by the quality control, there's also various different toolkits. So for example, there's FastX toolkit or Trimatic. So I know that Trimatic, is used in University of Manchester as in the core facility as a way to trim the adaptive sequences. So these kind of software allows us to discard low quality reads and eliminate poor quality bases from your results. So moving on to the profiling of your transcriptome. So first of all, you will want to do your read alignment. So you can map it either to genome, so whole genome that's available out there. So the softwares that can use is Top Hat and Star, or you can map it to just the transcriptome, which in this case you will use Bowtie. And if you want to do a reference-free assembly, i.e. if you're studying a species that's not very well known, then you can use Trinity to assemble your reads and then Bowtie to map it back onto your assembled transcriptome. And in terms of quality control, for the alignment, there's Picard, RCQC, and QualiMap. And for the count data quality control, there is RNA contamination detection or genomic DNA detection. There is the RNA-seq or EDA-seq. So in terms of kind of quantification and understanding like the differences, quantification at the genome level or at the intersample or intrasample level. For genome mapping, there is cufflinks. For transcription mapping, there is RSEM or Callisto. And for reference-free assembly, there is a hit-seq count or RSEM. So from this, you will then have a kind of list of raw reads that you generated from your RNA-seq results. But then what happens after the seq? So this is where you will start looking into the differential expression of the various transcripts available and then interpret your results from your analysis. So from your RNA-seq results, you will have a table of counts that are generated. So first of all, we will need to normalize it either between sample or within the sample. So within sample is usually we would use RPKM or FPKM. So FPKM, RPKM are essentially the same thing, but FPKM is fragments. So that is basically when you have pair end. So we have two reads on the same region. Whereas TMM is basically an additional kind of quality control so that you want to normalize it between the samples. So you first convert it into RPKM and then into TMM, depending on whether you need to look at intersample relationships. So after normalization, we will then have a differential expression testing. So this could be either done by Poisson or negative binomial testing. And for each of these, there are different types of packages that are available out there. So for example, DEG-seq or EdgeR, Bayes-seq, and DE-seq. So these are, so negative binomial tests will also involve Bayesian models. And so essentially it's basically trying to use a distribution curve to try and say whether or not your differences in the transcripts are with a normal distribution or not. So from these, you will then have a list of the differentially expressed genes, which then you can do systems biology approach and try and combine it with other types of data sets, for example, mass spectrometry analysis or actually DNA sequencing. And you can always test for whether certain categories or pathways are enriched using GoSeq. And then this should hopefully be able to generate some biological insight to answer what you're trying to find out. So the usual tools for this is for differentially expressed gene detectant is Lima, DESeq as shown here, EdgeR, SEMSES, RNAseq, and RNAseq Bio. So a very important thing to remember here is that if you do decide to do all these kind of analysis yourself, stick with what you're comfortable with. So these are all kind of algorithms are based on different languages. So if you're very comfortable with R, then try and stick with the package that's available for R, for example, Edge R, and instead of like trying to understand a new language, for example, Python. And always, if you're in doubt of your results, always talk with the experts. So there are not just experts that are around you physically, but there's also online forums whereby you can go and basically ask the question you want, and there will be tons of people that with more experience that will be able to give you a very detailed response and help you figure out what your problem is. So moving on to some common issues and also some pointers on how to resolve them. So the first thing is obviously, it's RNA-seq seems to be a very, there's a lot of things to think about. So usually people will say, oh crap, I don't know where to start. And my kind of go-to response is always, do you have someone that you can talk to? So in a lot of institutions, research institutes and universities, there's a core facility, and it's very likely that they already do RNA-seq analysis day in, day out. And if you don't have a core facility, you can probably find a group within your institution that is specialized in this technique. So essentially here, you really want to make sure that you have someone that is, that is very used to doing these kind of experiments to talk to. And if you do have a core facility or your collaborator has a very extensive amount of experience, they usually also have more than one option. So you should always ask what kind of packages they have available. And you also should discuss with them your biological questions. So because you need to decide whether you want to preserve your strand information, whether you want to do single-ended or pair-ended sequencing, and also how deep you want to go, and so on and so forth. Because all of these would have an impact on how expensive the package you're choosing will be, and also how complicated your downstream data analysis can be. So secondly, you need to ask yourself, what are you trying to study? So for example, if you're trying to study like a very straightforward genomic expression profiling, coding gene expression profiling, and you know that you have very good quality RNA, then you could just very straightforward and say, I want probably A enrichment. And if, for example, you're studying bacterial RNA, which doesn't actually have polyadenylation on their RNA, then you will have to use rRNA depletion or size exclusion. And the key point to remember here is that you don't actually necessarily need the most expensive package. So you don't need the biggest mean killing option. So don't go overboard. Just be very smart with your question and make sure you're picking the right package for the right kind of question. So another common issue that seems to arise is that qPCR results doesn't actually validate the RNA-seq data. So in this case, the first thing I always ask is your reference gene actually changing? So the reference genes you use in your qPCR, is it actually changing in your RNA-seq data, in your, for example, your conditional data set? Because very importantly, even though they're supposed to be housekeeping genes, they may still change. So for example, GAPDH is involved in glycolysis. So let's say if your experimental condition actually changes glycolysis, GAPDH expression might actually also change as well. So I'll always go back to your RNA-seq data and look up the usual housekeeping gene that you have and see if they're actually changed in your experimental setup. And in addition to that, I will always include a few other reference genes, for example, beta-actin, 18S, and RPLPO. Secondly, if you have replicates in your RNA-seq data, it's always good to go back and double check whether there is a huge variation between the replicates. So this can either be technical replicates. So if you have a huge variation between the technical replicates, then you have like an issue here, because essentially it means that something's gone wrong in your DNA prep or your RNA-seq sequencing. And so you need to go back to whoever is responsible for these different stages of trying to figure out where it's gone wrong. And if there is variation between the biological replicates, again, it depends on how big the variation is. But if there is, well, I will go back to the whole kind of variation between RNA-seq data, but this is something you need to take into account when you're trying to validate qPCR or validate RNA-seq with qPCR. And the third thing I will ask is, does your gene actually have splice variants? So certain splice variants might not be detected by your primers, depending on how you actually design them. And so you should go into your gene of interest and try and see if there are different exons or splice variants, and then design specific primers in order to see whether the RNA-seq data is actually specifically looking at these splice variants. And another thing is whether or not your gene is actually highly expressed. This sounds a little bit counterintuitive. However, too high expression might be problematic because it could be very hard to detect the differences amongst different samples. And finally, you can always go back to RNA-seq analysis and look at, first of all, what is your false discovery rate cutoff, so FDR. So if you have it set too low, then you will have a lot of false positive hits that is generated by RNA-seq data analysis package. And another thing is to make sure that the pipeline that is implemented by your bioinformatician, and to look at it and really understand what each step is and try and decide whether it's actually suitable for your study. So for example, in a paper in 2009, they actually found out that if you remove the untranslated regions from your RNA-seq data, it could improve consistency between your qPCR data and your RNA-seq results. And another thing to go back to RNA-seq results is that you can also just count the reads for the exons that you're specifically using for the qPCR detection and see if it correlates well with the qPCR. And remember that if you're trying to validate your RNA-seq results using qPCR, if you use the same RNA samples that you've used in the RNA-seq, then you only validate the technology. So you're just saying the RNA-seq basically is a good technology, and it's not actually adding any power to generating a good conclusion towards the hypothesis you might have based on your RNA-seq data. So always remember, use qPCR, use a different set of biological samples. So another issue might be that your RNA quality is consistently not actually good enough for, or not satisfactory enough for RNA-seq analysis, or indeed even qPCR. So the first thing is, I would ask, are you isolated from tissues or cells? So you need to make sure that you have a very optimized protocol for these tissue or cell RNA isolation. So cells is less of an issue because it's less complex. But for tissues, you might actually need to think about, maybe you need to digest the tissues with collagenase or elastase or dispase before you take the cells out and perform RNA isolation on those. And another thing is, it's always better to isolate RNA immediately after collection. So but on top of that, it's also better to perform RNA isolation in the same go. So obviously this is impossible if you have a time course experiment. So if you can't perform the RNA isolation immediately, then you should always store it in the RNA preservative. For example, RNA later that's available from QIAGEN, and you should always store at -80 until you're ready to perform the batch RNA isolation. So the general rule for RNA quality is always your RNA integrity number, or RIN, is higher than 8, and you want to ideally have your 260-280 and 260-230 ratios to be greater than 1.8. So for these, you can use nanodrop for the ratios, and we typically use Agilent bioanalyzer or tape station for the RIN analysis or RIN analysis. So you should keep in mind that even if your RNA quality is not good enough to match these guidelines, then you might still be able to analyze the data to get a good idea of your biological questions. So you should always ask the person who's going to do your RNA-seq what they think about A, your biological question, your sample, B, your samples, and see whether you can get away with using a slightly lower quality RNA. So another issue is that your RNA-seq repeat may not be consistent. So very importantly, we should always isolate the RNA using exactly the same protocol, and ideally actually using the same person, because every person has a different way of carrying out a protocol. So make sure you use the same protocol, and always make sure that you store it at -80 and no freeze thaw before your RNA-seq analysis. So every time you defrost a sample, there's always going to be a certain degree of RNA that is degraded due to the freeze thaw. And another thing is you can always check whether or not your RNA samples for RNA-seq analysis all have similar RINs and 260-280 or 260-230 ratios. So this will give you an idea of kind of sample variation as you're isolating your RNA. So key thing to remember here is that RNA are a lot more susceptible to degradation. And so you always make sure that you keep everything super sterile, make sure you have an environment that is DNase and RNase free, and work quickly, and very importantly, work on ice. So another issue that people usually want to ask is whether or not you want to mine. So you want to compare other data sets that are available online and compare it to what you've done so far. So the first thing I will always go for is you should check whether or not the authors have a spike-in standard that you can actually use in order to do a direct comparison between their data sets and your data sets. And secondly, you should try and figure out if their RNA quality is good enough for a direct comparison as well. So this is a key issue that is still very prevalent now in terms of publishing RNA-seq data, because there's a lot of missing metadata that makes it very hard to exclude bad RNA-seq results, or sorry, not results, but rather samples, or at least allow you to have a kind of idea of whether or not you should implement different computer algorithms to account for example, RNA degradation. And about 2 years ago, there is a paper that was published in Nature Communications, whereby the authors have designed a relatively new tool called mRNA, which is available for free. So you can directly assess mRNA integrity using this mRNA tool. So I've not used it myself, but I have heard good feedback from other people that this is a tool that's worth looking into. And key thing here is to always remember that the bioinformaticians, the core facilities, your collaborators, people who have actually done this day in, day out, are here for a reason. So anytime you have any questions, you should discuss with them. So moving into the final kind of bit of my presentation. So essentially, in my humble opinion, the best of both worlds, i.e. minimal cost and maximal effect, which is something I love doing, is to have the RNA-seq and probably a proper analysis of the RNA-seq data combined with qPCR. So how do you do that? You can actually have a RNA-seq and the data analysis on, let's say, 2 or 3 biological repeats, and make sure you have good and competent bioinformatics analysis. And then you can then generate a list of differentially expressed genes, which then you can implement in your qPCR validation. So in this case, you don't actually even need to go through the whole list of the DEGs. You can just pick a few key ones that you think are very interesting and validate your conclusions in as many biological samples as possible. So in terms of practical tips before you kind of go on this journey of RNA-seq, you should always think about your biological question. So what are you actually trying to find out? So whether or not your question is actually justifiable to use RNA-seq in terms of the costs. And if you do decide RNA-seq is a way forward, which library prep you should use, and which, do you want this just one-ended or pair-ended analysis? And secondly, you need to think about how much power do you need in your RNA-seq analysis. So for example, if you know that you're going to study a very well annotated genome, then perhaps you don't need to go as deep as you think you need to. And finally, you need to figure out who's going to do all the different steps of the RNA-seq. So for example, who's going to isolate the RNA, who's going to prepare the cDNA library, and who's going to run the actual sequencing machine, and finally, who's going to analyze the data and make sure you're in good communication terms with every single step if you're not the one that is doing everything, which I really highly suggest you not to. So very important thing is to always plan. So I can't stress this enough. So you need to make sure you have enough time beforehand to plan, to think, to practice, and so that on the day of the actual experiment, you're not panicking. You want to make sure that you are relatively stress-free. And another thing is to make sure you have a very sterile RNA workspace. This is, again, a very important key issue in doing both qPCR and RNA-seq, in fact. So you want to make sure that all your tips and reagents are DNase-free and RNase-free, and always remember to perform RNA quality check, i.e. the RIN number should be greater than 8 and the ratio should be greater than 1.8. And one key thing here is that you need to remember that the RNA that you isolated, you always need to have a certain amount reserved for this quality check stage. So bear that in mind when you're isolating RNA and try and accommodate for that loss of RNA that is used in these adjuvant or nanodrop analysis. And finally, it's very, very important to have good communication with your technicians and your bioinformaticians and your core facility and or your collaborators. And at the end of the day, if you're unsure, you should always ask someone else who's experienced. There's no stupid questions, especially in science. Always ask if you're unsure, and don't try and push through an experiment if you're unsure about any part of the steps that you need to do. And here are some useful tools, kind of online resources that I always go to for both qPCR and RNA-seq. So I tend to use NetPrimer to check how good my qPCR primer sets are. So this would give you a kind of a score out of 100, 100 being the best, and also it will give you kind of a reading of how likely your primers are forming primer dimers under your experimental conditions. And secondly, this Scotty, which is an algorithm that is developed by University of Utah, you can use that for your experimental design. So essentially, this is a tool that you can decide how much power you need, so how many replicates you need, and how deep you might want to sequence your data. And the MRIN tool that I mentioned briefly before is available in omictools.com, and the GO2R, which is available through the NCBI website, is a quick tool that you can use to mine publicly available datasets from GO. And it's a very user-friendly interface, and you can actually look at the gene-by-gene basis for the different RNA-seq data that might be available out there for a gene of interest. And finally, seekanswers.com and bioslides.org are both kind of forums that are available for you to post your question on there and have experts explain both. It might be theory or it might be kind of actual guidance on which steps you should implement in your data analysis. And in terms of reviews, the review by Conessa et al. That I briefly used their kind of roadmap for earlier in this presentation, I think is a fantastic introduction level of RNA-seq analysis for those who are beginners. And there's also a comprehensive assessment of the RNA-seq methods by the SEC-C consortium that is published in Nature Biotechnology in 2014. And if you're interested in more in-depth bioinformatics comparison, then there is a relatively new kind of survey on the bioinformatics that are available out there for specifically RNA-seq data analysis, and also a free BMC bioinformatics paper that's comparing different methods. I believe in this case, they're comparing 11 different methods for the differential expression analysis for RNA-seq data. And finally, there's also Bite-Size Bio, the qPCR webinar, and also various different blogs that they have published before that will give you various tips on qPCR, multiplex PCR, and also RNA-seq. So finally, the take-home message is always remember that having good samples are only the beginning. In order for the good samples to come through, you also need good data analysis. And finally, always the 3 steps is plan, ask, and keep calm, and keep asking. So thank you all for listening. And yeah, I will take any questions now, Karen.
Thank you, Joan. That was a really excellent presentation. We do have a few questions from the audience. If anyone else has a question at this time, please feel free to post it in the questions box that appears on the right-hand side of your screen. So the first question up is from Amit, and Amit also says, thank you, Joan, for a very nice, informative presentation. This is a very good question. I don't know if you can answer it, but what do you do if there's a variation between RNA-seq and qPCR results and you cannot resolve it with the tips that you presented? Is there any other method that you can bring in to try and settle the score?
Right, so you're having issues. So basically you're saying that your gatekeeper is not changing and all the different tips I've
it's tricky, right?
It is tricky. I'm just trying to think if so, I mean, my first question would be like, is he trying to kind of validate his RNA-seq data using the same RNA samples, or whether it's a different biological sample? Because at the end of the day, if you're trying to validate using the same RNA sample and you don't get anything, like, you don't resolve the differences between RNA-seq and qPCR, then you're basically in a very bad situation. So that's the first thing I'm thinking, is to try and kind of figure out whether there's an issue with the RNA-seq analysis that's gone wrong, or maybe it's a qPCR that, has he actually looked at the primers, efficiency, binding, and, kind of going back to the whole melt curve and understanding how good your primers are actually recognizing your gene of interest in this case, I guess. I mean, well, and also going back to your bioinformatics person, it might be himself, and going through the RNA. I'm guessing, and also, well, another thing is, I would like to know whether he is using a de novo assembly, because if you're mapping your RNA-seq data back on a kind of like a self-generated genome like mapping, then there are certain things that you need to kind of keep in mind. So for example, you need to make sure that the adaptive trimming is carried out correctly so that you can remove contamination from that. And this is especially important for de novo assembly, because if you have some traces of adapter left over, then obviously that's going to give you a genome to map out your reads that's incorrect. Yeah.
Right. Yeah. So I guess really the take-home message is that if everything has been done correctly, then qPCR and RNA-seq should actually be in agreement with each other.
Yeah. I can't really think of anything particular reason why it shouldn't be.
No, I can only think of a northern blot, but that's kind of going back in time instead of going forward.
Yeah.
So yeah. Okay, that was a good question. We also have a question from Bharat. So you mentioned something about a spiked-in control. Can you elaborate on that? And are you aware of any commercial kits available for this, for RNA-seq?
I have to say, so like spike-in control essentially is kind of like it's like a known amount of sequences that you put into your sample. So it's basically trying to, using a known amount of transcripts. So this will be a spike-in, and then comparing your different transcripts in your actual sample to this kind of baseline that In terms of commercially available, I'm pretty sure there is plenty of those out there. I can't really comment on that myself right now, because I've never done like the whole kind of spike-in myself. But there are tons. I'm pretty sure there are tons of commercially available spike-in controls I can use. So the first thing I would go maybe is to Illumina platforms and look at different like internal controls that they might have.
Yeah, that's fair enough. We also have a question from Sandra. So we didn't really talk much about housekeepers in the presentation, but do you have like a number of housekeepers that you would recommend?
What does she mean in terms of how many for the qPCR? Like, so generally, how many you should do for each qPCR?
I guess so. I mean, you mentioned that. You also mentioned that the housekeeping genes that you would analyze in qPCR may behave differently in RNA-seq.
Yes. Yep.
So I guess the question is, how many housekeeping genes in qPCR would you need? I know it's like the holy grail question, but what is your feeling?
The first thing is to, I would say the first thing is to like look at your biological, like your different experimental group, and try like figure out whether there is a reason why your current gatekeeper is not working. But I would say, so in my own experience, like I would always use maybe 2 or 3 to start with when I'm optimizing, just as like a quick look see when I'm doing the experiments, to see if it's changing with my experiment condition. And that's even before I look at my gene of interest. So I would go like 2, 3 generic ones, and then if those don't work, then I would go for, additional maybe 2 or 3, just to make sure that, I have 1 or 2 good fallbacks on. And then from then onwards, I would usually just use 1 gatekeeper, because I've already done the optimization and I know that these don't change in my experimental conditions.
Yeah, that's great. And so when you say that the housekeeping gene could change in RNA-seq, why would that be? Would it be because it's more sensitive, or why might that be?
No, I mean, I think it's actually more of a case of your experimental condition might actually be changing it without you realizing it. Because a lot of people, when they start, for example, PhD students or something, I have this mistake myself as well. Like, I think the beta actin is always expressed at the same level. There's no way it's going to change its expression. But obviously, there are very vast amounts of experimental conditions that can actually change the expression. And I mean, so going back to the qPCR, if you go back and have a look at your gatekeeper and you realize that they have very different cycles, they come out with very, very different cycles, then that's already a red flag that maybe your kind of experimental samples are changing how it's behaving. So I think it's more of a kind of like the experimental setup rather than the sensitivity of RNA-seq.
Yeah, so if you could trust your housekeeping gene, you could actually use that almost as a way to see how close your sample was likely to resemble your qPCR sample.
Yeah, to an extent, yes.
Yeah. Okay, good. We also have a question from Hisham, who says that RNA-seq will determine messenger RNAs, microRNAs, and what other kind of RNA species?
Oh, you have
everything, I guess.
Everything. So you can have long non-coding RNA, short non-coding RNA. You can also have, I'm pretty sure they can do tRNA, because RNA-seq is very good at detecting secondary RNA species as well. So there are tons. Basically, I think. Well, I don't want to give a completely kind of 100% sure answer, but basically any type of RNA that you can.
Yeah, I guess.
Yeah.
It also depends on the quality of your RNA sample and how good your RNA extraction technique is to get the really small RNAs out, so on.
Yeah, but I mean, I guess one thing is, one thing that I always need to think about is it also depends on your sample. So, for example, if you have a sample that is FFP treated, then your RNA is probably going to be degraded. So you need to keep in mind that even though you use a particular package, the degraded mRNA might still trickle through. So, for example, microRNA, maybe you're using specific microRNA kind of size selection, but then because the degraded mRNA, they're shorter, they would then come into your actual sample pool.
Yeah, that makes sense. And I think we have time for one more question before we wrap it up. This is from Kriti, who asks, what does it mean when the FPKM value is 0 for a gene in my sample? And in case you're not familiar with the abbreviation, it stands for the fragments per kilobase of transcript per million mapped reads. So the question is really, when the number of fragments per kilo
is 0.
Is 0, what does that actually mean? Does it mean
it means
transcript?
It could mean that, but then it could also mean that your transcript is very, very low level. And this is particularly an issue when you're trying to do kind of a fold change between samples. So it's always better to have a cutoff for RPKM at a certain level. So at least maybe even 2 or 3, I would say. Because obviously, if you have something that's 0, then you get an infinity fold change, which is a bit hard to interpret.
Yeah.
So I mean, it depends on how deep you're sequencing. So, for example, if you have an RPKM of 0 for, let's say, you've gone 1 million kind of base reads, then you not base reads, but you basically you're going oh, sorry, yeah, a billion, sorry, not 1 million, but a billion, then chances are you probably have no expression of that transcript.
Right.
Yep.
Okay. Thank you, Joan. So that brings us to the end of the seminar. Thanks again, Joan, for a very illuminating presentation and a really fantastic discussion.
Thank you.
Thanks also to our sponsor, QIAGEN. And finally, thanks to you, the audience, for taking the time to attend and listen in. If you have enjoyed the seminar and would like to view the video recording of the session, please visit the seminars page on bitesizebio.com. It should be available within the next 24 hours. There you can also see the other webinars we have lined up for you in Bitesize Bio's Webinar Festival. So until next time, good luck in your research and goodbye from all of us at QIAGEN and Bitesize Bio.